Our research project focuses on the dietary impacts on development time in fruit flies, which are also known as Drosophila melanogaster. The project members consist of Emma Ferlin, Priya Gupta, Benjamin Kurtz, Grace Callahan, Angela Wharton, Courtney Nave, and Justin W. Mary. This project is completed through the Department of Biology at St. Francis University, located in Loretto, Pennsylvania. First, we will begin with some background information about how this research project came about. From previous research, there has been a rather broad and ongoing debate on nutrients that impact the growth of sensory structures. For this project, Drosophila melanogaster was used to test the hypothesis that dietary impacts affect the growth of sensory structures. Drosophila melanogaster was used in this project because they have fast larval growth on artificial diets, and their diets are easy to manipulate. From the start of this research project, our team collaborated and introduced two quite specific hypotheses. The first hypothesis was that if the development in flies is energy limited, then their development will be severely impacted by the low carbohydrate diet. On the other hand, if the development is nitrogen or phosphorus limited, then their development will be severely impacted by the low protein diet, but not the low carbohydrate diet. Once again, our hypotheses strictly focused on how the specific diets affect the development in fruit flies. The picture on the right shows the trays of our fruit fly diets in the environmental chamber. Like previously stated, in the fall of 2021, there were 3 diets made in smaller batches. Each batch contained at least 20 vials of food, and there were 5 batches of each diet made over the course of a few weeks, totaling to a minimum of 100 vials per diet. This was done to ensure that we would still get some offspring in case one batch of food was bad, and also to split up general emergence times so that there would not be an overwhelming number to collect at one time. The 3 diets still consisted of a normal, low carbohydrate, and low protein. The general plan for the methods was to make the food, load the parents, at least one female, unload the parents when larvae started to appear, and then collect the offspring daily. Our fly pushing setup can be seen on the right side. These were the exact measurements for our 3 diets. The normal diet plan was based on the one from Sedella Blatch, and was modified to fit our experiment for the other 2. We used cellulose, which cannot be digested, to replace the dry ingredient mass on the 2 experimental diets. Drosophila do not have genes for cellulase. On the right side, the general differences in nutritional value between the diets are shown. Spring of 2022 marked the beginning of our measurement portion of the project. The flies were grouped together based upon their specific batch, which was 1 through 5, and their diet type, which was normal, low protein, or low carbohydrate. A total of 8 families, if possible, from each diet and batch were sexed. Then they were weighed in one big group based upon sex. In general terms, all of the females were weighed together from that batch, and all of the males were weighed together from that batch. Next, the sexed and weighed flies were measured by thorax length. A total of 5 males and 5 females were measured from each of the sexed families. The general concept for how to measure the thorax of a fly is in the picture shown here, where the yellow line is the length we would take for our data. The eye measurements are currently in progress, and we hope to have that data soon. The graph on the left shows the average development time of the flies on the 3 different diets. The y-axis represents the development time, which is in days. The x-axis represents the 3 different diets. The flies that were raised on the low-protein diet have a significantly longer development time compared to the flies that were raised on the low-carbohydrate or normal diets. The p-value that was calculated for the development time of the flies from the low-protein diet as compared to the other 2 diets is less than 0.01. This suggested that there is a significant difference in development time between these diets. The graph on the right shows the average number of flies per family across the 3 different diets. The y-axis of this graph represents the average number of flies per family. The x-axis represents the different diets. All the diets were significantly different from each other. The p-value that was calculated was less than 0.05, which suggested that there is a significant difference between the number of flies that developed on the different diets. Here, you can see mass and thorax length across our 3 diets. As with most insect species, and as is typical for fruit flies, females were consistently larger than males. In mass, we did see a small but significant difference between the normal diet and the low-carbohydrate diet, which was that low-carbohydrate flies were larger. We also found a significant effect of diet on thorax length, but we suspect that finding is caused by heterogeneous variance more so than a difference in means across diets. Overall, we have found that low-protein flies showed longer development times and a lower amount of flies per family. Nevertheless, those flies that did eventually emerge from low-protein diets were not substantially different in size than those of the other diets. The low-carbohydrate diet surprisingly led to more surviving flies, but similar development times and body size compared to the normal diet. Therefore, our data support the hypothesis that flies are protein-limited, not energy-limited. This is likely due to either nitrogen or phosphorus limitation, or possibly both. Furthermore, the fact that low-protein diet yield comparable body sizes despite apparently stressful conditions may indicate that the flies compensate for the stressful diet conditions by increasing development time to yield the same sized animal. Our future plans involve investigations of eyes, which are composed of nervous tissue and have a very high metabolic rate, estimated at 10 times that of the average tissue. Through measurements of the eyes of our animals, we will test the hypothesis that eyes are more sensitive to energy limitations than other structures. We would like to thank St. Francis University for the use of their laboratories and for the research grant. We would like to thank Dr. Mary who helped to organize this research project and supervise. We would also like to thank Sidella Blatch for the use of her normal diet recipe. Finally, here are references that were quite useful in the making of this project.